Hi, good afternoon. In this session, I am going to discuss electron shuttle systems which are present in eukaryotes. Actually, these shuttle systems are absent in prokaryotes because in prokaryotes there are no membrane bound cell organelles. So, shuttle systems are absent. On the other hand, electron transport system of the prokaryotes takes place on mesosomes which are in contact with the cytoplasm. But in the case of eukaryotes, glycolysis takes place in cytoplasm, Krebs cycle takes place in mitochondrial matrix, whereas the electron transport system takes place on inner mitochondrial membrane. Here the NADH2 which are produced in glycolysis should enter into the electron transport system to undergo oxidation resulting in the formation of ATPs through oxidative phosphorylation. But inner mitochondrial membrane is impermeable to NADH2 that are produced in cytoplasm. NADH2 which are produced in cytoplasm are said to be cytosolic NADH2 or cNADH2. So cNADH2 cannot cross the inner mitochondrial membrane, but to involve in electron transport system, they should enter into the mitochondrial matrix, but they cannot. So directly NADH2 cannot cross the membrane and enter into the mitochondrial matrix, but NADH2 transfers its reducing equivalents to either FAD plus or NAD plus, reducing them into either FADH2 or NADH2. So that is why while discussing the electron transport system, I said that the number of ATPs differs in the case of cytosolic NADH2. Sometimes cytosolic NADH2 yields 2 ATPs, sometimes it yields 3 ATPs. So that depends on the different shuttle systems, electron shuttle systems which are present in only eukaryotes. So in this session, let us discuss the two shuttle systems. There are two shuttle systems known malate aspartate shuttle and glycerol phosphate shuttle, which I have written in blue ink, malate aspartate shuttle, and here it is glycerol phosphate shuttle. So malate aspartate shuttle is present in liver cells, kidney cells and heart cells. And this is more efficient shuttles in which high number of ATPs are formed. Whereas in the case of this glycerol phosphate shuttle, it is less efficient. There, as compared to the malate aspartate shuttle, low or less number of ATPs are formed. And this takes place in skeletal muscle cells and brain cells, whereas that malate aspartate shuttle takes place in heart cells, kidney cells. And coming to the malate aspartate shuttle, here in the malate aspartate shuttle, NADH2 transfers its reducing equivalents to the oxaloacetic acid. NADH2 transfers its reducing equivalents to oxaloacetic acid, so that oxaloacetic acid is reduced to malate, while the NADH2 is oxidized to NAD+. Actually, the NADH2 which is produced in glycolysis transfers its reducing equivalents to oxaloacetic acid that results in the formation of malate. So, oxaloacetic acid is reduced to malate. Simultaneously, NADH2 is oxidized to NAD+. So, now malate is resulted in the cytoplasm itself. Now, the malate can enter into the mitochondrial matrix because the mitochondrial inner membrane is permeable to the malate. Now, malate inside the mitochondrial matrix, under influence of malate dehydrogenase enzyme, it loses reducing equivalents which it received from the NADH2. Actually, NADH2 transferred its reducing equivalents to oxaloacetic acid, so that oxaloacetic acid is reduced to malate. Now, from the malate, the reducing equivalents are removed under influence of malate dehydrogenase and the same are added to NAD+, reducing the NAD+ to NADH2. and malate will be oxidized to oxaloacetic acid again. So here cytosolic NADH2 in the case of malate aspartate shuttle, it transfer its reducing equivalents to again NAD plus. Hence here also NADH2 is resulted. Now here oxaloacetic acid is formed now. This oxaloacetic acid cannot cross the inner mitochondrial membrane because again the inner mitochondrial membrane is impermeable to oxaloacetic acid. It does not allow the oxaloacetic acid to go out. So oxaloacetic acid undergoes amination, resulting in the formation of an amino acid known as aspartate. Now aspartate can cross the mitochondrial inner membrane because the mitochondrial inner membrane is permeable to the aspartate. So aspartate comes out, and through the deamination again it forms oxaloacetic acid. So like this, what happened here? Nothing. Just NADH2 transferred its reducing equivalents to NAD plus. So that again NAD plus is reduced to NADH2. So in this case, malate aspartate shuttle, even inside the matrix also NADH2 only formed. So when malate aspartate shuttle is operated, whether how many ATPs are formed, let us calculate. Coming to the glycolysis. In glycolysis, 2 NADH2 are formed. Now 2 NADH2 into each NADH2 is equal to 3 ATPs. Because the cytosolic NADH2 in the case of malate aspartate shuttle resulted again in NADH2 itself, even in the matrix also. So that gives 3 ATPs. 2 into 3, that equal to 6 ATPs. 2 into 3 equal to 6 ATPs. And however, 4 ATPs are formed directly through substrate level phosphorylation in the glycolysis. So 6 plus 4, 10 ATPs. From 10, 2 are invested in the glycolysis for the phosphorylation of the glucose to glucose 6 phosphate and fructose 6 phosphate to fructose 1,6 bisphosphate. So 10 minus 2, 8 ATPs is the net gain in the case of malate aspartate shuttle. And coming to the link reaction, there are 2 NADH2. Each NADH2 gives rise to 3 ATPs. 2 into 3 equal to 6 ATPs. Coming to the Krebs cycle, there are 6 NADH2. We know where they are produced. 6 into each NADH2 equal to 3 ATPs. 6 into 3, 18 ATPs. And 2 FADH2 also there. Each FADH2 is equal to 2 ATPs. That equal to 2 into 2, 4 ATPs. And in Krebs cycle also, through the substrate level phosphorylation, 2 ATPs are formed. So 2 ATPs. Together, in a glycolysis, 8. In connecting link, 6. You calculate connecting link and Krebs cycle. 6 ATPs from connecting link reaction, and here 18, 6 plus 18, 24 plus 4 from FADH2, 28 plus 2 through substrate level phosphorylation, 30 are resulted. In a glycolysis, 8 net gain, 30 plus 8, 38. So total number of ATPs produced in malate aspartate shuttle. If malate aspartate shuttle is operated in the aerobic respiration, total number of ATPs are produced are 40 minus consumed 2, and the net gain is 38. So what is the net gain of ATPs in the case of malate aspartate shuttle? 38. What is the total number of ATPs formed in aerobic respiration if malate aspartate shuttle is operated? 40 minus 2, 38. Like this, in one case, cytosolic NADH2 is equal to 3 ATPs. Now let us discuss another case where cytosolic NADH2 equal to 2 ATPs. So that is in the case of glycerol phosphate shuttle. Here what happens? Actually it takes place in brain cell and skeletal muscle cells, and it is less efficient. Less number of ATPs are formed as compared to the malate aspartate shuttle. Here what happens? NADH2 transfers its reducing equivalents to DHAP, dihydroxyestrogen phosphate, which is a triose phosphate. By transferring the reducing equivalents to DHAP, NADH2 is oxidized to NAD+. Due to transfer of reducing equivalents to DHAP, dihydroxyestrogen phosphate is reduced to glycerol phosphate. Now the glycerol phosphate enters into the perimitochondrial space. It goes up to the outer surface of the inner mitochondrial membrane. There, it transfers its reducing equivalents which received from that NADH2 to not NAD plus but FAD plus. That is the difference here. In the case of malate aspartate shuttle, the reducing equivalents are transferred to NAD plus so that NADH2 resulted. But in the case of glycerol phosphate shuttle, reducing equivalents from NADH2 are transferred to FAD plus so that it is reduced to FADH2. Due to transfer of reducing equivalents to FAD plus, glycerol phosphate is again converted to dihydroxyestrogen phosphate. The dihydroxyestrogen phosphate from the perimitochondrial space, it crosses out the outer membrane and goes into the cytoplasm. So now here in a glycerol phosphate shuttle, FAD plus is reduced to FADH2. Whereas in the case of malate aspartate shuttle, NAD plus is reduced to NADH2. So in the case of glycerol phosphate shuttle, the cytosolic NADH2 transfers its equivalents, reducing equivalents to FAD plus so that is reduced to FADH2. Means one NADH2 equal to an FADH2 here in the case of glycerol phosphate shuttle. If it is FADH2, FADH2 gives rise to only 2 ATPs. Okay, now let us calculate whether how many ATPs are formed in aerobic respiration if the glycerol phosphate shuttle is operated. You see here, in the glycolysis 2 NADH2 are there. But now these 2 NADH2 are equal to FADH2 because NADH2 transferred its reducing equivalents to FAD plus so that FADH2 are formed now. So here each NADH2 gives rise to only 2 ATPs because FADH2 formed now. 2 NADH2 into 2 ATPs equal to 4 ATPs. And however, through SLP, substrate level phosphorylation in the glycolysis, 4 ATPs are formed. 4 plus 4, together 8 ATPs. And to our invested now, for the phosphorylation of the glucose to glucose 6-phosphate and fructose 6-phosphate to fructose 1,6 bisphosphate. So 8 minus 2 equal to 6. The net gain of ATPs 6 only in the glycolysis. And in the Krebs cycle and connecting link reaction as usual, same. In the linked reaction, 6 ATPs are formed. In the Krebs cycle, 18 ATPs are resulted from NADH2 and 4 ATPs are resulted from FADH2 and 2 ATPs are directly produced through substrate level phosphorylation, together 30 plus 6 ATPs in glycolysis. So totally we get 36 only. Total ATPs formed 38 because 30 in connecting link reaction and Krebs cycle together and 8 in the glycolysis itself. 30 plus 8, 38 minus how many ATPs are consumed in the glycolysis? 2. So 38 minus 2, that equal to 36. See, if it is malate aspartate shuttle, 40 ATPs are formed. Minus 2, net gain becomes 38. But in the glycerol phosphate shuttle, total number of ATPs 38 minus 2 consumption and net gain becomes 36. So where it is a difference? In the connecting link reaction and Krebs cycle, same. Whether it may be malate aspartate shuttle or the glycerol phosphate shuttle. In both the cases, same number of ATPs, 30 formed. Difference is in glycolysis. If it is malate aspartate shuttle, there 6 ATPs are formed from the cytosolic NADH2. If it is glycerol phosphate shuttle, there only 4 ATPs are formed through the NADH2, cytosolic NADH2. Hence, the net gain differs, even the total number of ATPs also differs. If you see the malate aspartate shuttle, it is high efficient because here total 40. But if you see the glycerol phosphate shuttle, here only 38 are formed, means it is less efficient.